here we're going again. Okay. So as I said, ours isn't on a glass floor, so it's not quite as attractive, although it looks very similar. So our new magnet arrived in November. So this is a picture of it coming off of the actual semi. So it was built in Germany and then flown to the United States and actually set on a magnet farm in Chicago, where they gave it vitals for about a month as we finished the location where it would be installed. Literally a bunch of guys actually pulled it in. It weighs 28,000 pounds, so that was actually really fun to watch. Then they got up to the door and it wouldn't fit, which we knew it would be really tight. So then they had to take all the housing off the side to pull it through, which was actually really neat. So you can see actually what it looked like underneath all of the plastic housing. So then took a whole day and they got it into place. And then this is what it actually looks like today. And it's in the Hickson Lead Center. So basic way it works. So patients come in, they lie on this bed, they go into the bore. Then of course in the bore, there's a very homogeneous magnetic field that lines up protons well, basically all the protons in your body with the actual field. And then there's of course part of the inside the bore that's stronger than the other parts, and that's where you actually want the organ that's being imaged to be located. And then behind the housing, there's gradient coils that disturb the protons, and then in MRI you're measuring their decay back to the baseline. And you can do that a lot of different ways depending on what you actually are trying to image. So a few details about our magnet. So it's the Prisma 3 Tesla model, which is considered the research model. It has advanced gradients. It comes with a smaller bore, so it's 60 centimeters, whereas most clinical magnets are 70 centimeters. So that's good in the sense that the coils are closer to the body, so you can get a much more crisp image. It's bad in the sense that it's a little more claustrophobic than a standard clinical magnet. Came with 3 head coils. So there's a 20-channel coil, 32 channel, and a 64 channel. 64 channel is state of the art. And basically on a head coil, the more receiving coils you have, the faster you can acquire data. And to some degree, the better coverage you have depending on where those actual receiver coils are located. It also came with a more high performance computing system on the acquisition side, so you can acquire data very quickly. The model we got was equipped with all of the available commercial sequences and applications, so it's basically loaded with anything you'd want to do. And the overall facilities operate as a research core, and it has a dedicated research technologist and a manager. And as I mentioned, it's located in the Hickson Lead. And if you're familiar with the MRI systems at UNMC, this is the same location where the 3 Tesla GE scanner sat for more than a decade. Just been renovated significantly, as you saw. Of course, we also have a Magnetoencephalography, or MEG system. This system was installed in very late 2008, early 2009. This is an image of it. So the way this system actually works, patients come in, it's in a huge magnetically shielded room that's decorated to look like it's not a magnetically shielded room, but in reality, when you're in here, you're like inside a bank vault. So you walk through a door that's over here on the side, have a seat, and then the seat goes up and the person's head fits within the helmet. And then you can kind of see there's little trays here. So once they're all positioned, a tray slides in and their arms rest on that tray. And then there's a screen that's wheeled in front of them where you can present stimuli and other devices. The instrument also rotates where it can be put into the supine position. And then this is actually a bed here that's wheeled in front, so you can do recordings both seated and supine, depending on kind of what your goal is. Inside the helmet, there's a large dense array of sensors that measure very tiny magnetic fields. And so that's really what you're doing in MEG, is measuring magnetic fields naturally generated by the brain. A little bit how the system works. So as I mentioned, there's an array inside the helmet. In this big area here, there's a dewar hidden behind the housing, and that dewar is full of liquid helium. So literally a couple of centimeters from the scalp surface, it's negative 452 degrees Fahrenheit. And all of the sensors that are measuring those magnetic fields are all being bathed in that. So that gives them superconducting properties and allows them to measure magnetic fields on the order of femtotesla, which is about 1 to 10 billion times smaller than the Earth's steady state field. So a few facts about the system. Even 10 years later, it's still state of the art. It has 306 sensors, which is still the most available in any commercial system. 204 of those are planar gradiometer coils, so they're ideal for measuring cortical activity. So just the very top thin layer of the brain. 102 of the coils are magnetometer coils, and so they're designed to measure deeper structures, so basal ganglia, thalamus, deeper structures going down really, it's theoretically as deep as the brain goes. System is equipped with a smart shield, which is in 2009, we were the first people to actually ever use the smart shield. So it's basically you're sending electric current into the walls of our magnetically shielded room, and it creates these huge compensation fields, which is a very advanced way of doing noise reduction. Because if you think about it, so we're measuring these ultra tiny magnetic fields. So they're hidden in this magnetic chaos of a hospital. And so we have to block out cars and electrical lines and computers and people and doors and basically everything. And so that's the kind of the reason we have the smart shield. We also have software and hardware for continuous head tracking and motion correction. So we were the first site 10 years ago to actually implement this in research studies. So we can monitor the head in real time as a person's, even if a person's sitting still, they're still breathing. They're still, if they're a child, they're not really sitting that still. If they're an adult, they're probably not sitting that still either. And plus they start to slouch throughout the recording because they're sitting up. So we're able to track that in real time and then correct for it, as long as it's not just, an enormous amount of movement. We can sample the full array as high as 6 kilohertz. So that means taking a measurement at each of the 306 sensors at tens of microseconds if we really needed to. Like the MRI, the MEG has a dedicated technologist, and it's actually located, unlike the MRI, in the South Doctor's Tower on the 2nd floor. So a few words about just research applications, both the systems. So start with MRI. So one of the things that a lot of people are doing really across the world are doing functional measurements using MRI called fMRI, and then doing resting state or task-based network analyses. So probably one of the latest, I wouldn't say the latest, I'd say the most popular thing you see currently in cognitive neuroscience is resting state studies using fMRI. So in this, you acquire, the person just lies in the scanner, you acquire fMRI data while they're usually sitting there with their eyes closed, and then you can do these network analyses based on there's a bunch of different varieties. One of the most common is independent components analysis, or ICA. And what that does is it extracts these intrinsic functional networks, and then you can do advanced analyses and look at how these networks talk to each other. So this is kind of an example here. The way they're coded here is like, so this is auditory. You can see they're in the auditory cortex, and there's essentially 2 that are highly correlated, and then 2 others shown in blue and shown in red. And then you can also go into the sensory motors. These are groups of brain regions that are their activity is correlated, and so therefore they're theoretically a network. And then you can, using advanced techniques, look at the correlation or interactivity between the different networks. So you can look at, you localize, a network in sensory motor cortex, and you can look at how activity in that network is correlated with other brain networks that are across the brain. So essentially, using these techniques, you can look at, how many networks are there in the human brain while it's at rest. So let's say there's 30 different networks, and then how are those networks each talking to each other? Then how are the individual brain regions in each of those networks also talking to each other? And so basically using our scanner, many others, you can compute this. This is actually just one looking at healthy controls, but you can do it, look at how networks are different in patients, or look at how they're different in, with progression of a disease, or really anything. Scanner also does what's called diffusion imaging. So what this is doing is, I should have said, so fMRI, basically you're looking at changes in the hemodynamic signal, which is the oxygen versus deoxygenated ratio of hemoglobin. That's where you get your contrast. The way of just thinking about it, though, is that you're essentially based on how much blood flow is going to that region. So regions of the brain that are active have increased blood flow to them, and then there's a special way you can extract contrast from that and get a signal that's indicative of function. Diffusion is a little different. So you're looking at the diffusion of water molecules. And the main thing this is currently being used for is looking at white matter tracts in the brain, white matter pathways. So we'll start here with the spinal cords. This is a really pretty one, as you can see it. All the spinal cord tracts, all the spinal cord fibers are headed in the same way. If you've had neuroanatomy, you know, like in the spinal cord, there's lots of nuclei where everything in that nuclei is extremely organized and go into a specific limb or a specific part of the body. So all those are perfectly organized. You can pull that out and see it in diffusion imaging. Then going into the brain, you can look at pulling out, you know, the major tracts and looking at their actual direction in the head. And that's what's shown here. So like in red, you're looking at fibers that are going from left to right. So you can see like the corpus callosum here. And this one, and of course these colors are just added to it to illustrate where the actual fibers are going. And blue in this is fibers that are going superior to inferior. And then your green fibers are, that's right. Yeah, going anterior to posterior, I believe. Yes, so you can see it better here. So you can see the green fibers are going anterior to posterior, and then red's going left to right, and then blue's going up and down. So essentially the colors are just added to, again, show which the actual direction of the fibers are. We can also do spectroscopy. So here, spectroscopy allows you to look at different metabolites. So these metabolites may, you know, be related to inflammation or different things. Depending which one you're interested in, there's usually quite a bit of literature where you can focus on one in particular. And then based on the size of the voxel, you can have better or less resolution. Bigger voxels, you typically get better signal to noise ratio, so you have better signal. On the other hand, big voxels are usually less meaningful because everyone's interested in a smaller brain area or a specific target. And so the voxel gets smaller, but at the cost of the signal. One thing we're particularly interested in here is looking at GABA. A lot of diseases are thought to be inhibitory disorders. And so looking at local GABA concentration and correlating that with disease parameters is a major interest. Perfusion is another thing that our scanner can do right out of the box. So here you're looking at quantitative blood flow measurements to different areas of the brain. This is a whole brain measurement. So like here, I'm just showing one slice, but you actually, unlike spectroscopy where you only have the voxel, you have a full brain measurement here. And so like in this map, what we're looking at is there's quantitatively more blood flow to multiple brain regions, especially the frontal lobe in younger healthy adults versus older healthy adults. And so one of the nice things about perfusion is that it's quantitative, unlike most MR-based measures. So you can look at you have actually a quantitative metric that's continuous that is really mathematically friendly. Susceptibility weighted imaging is another thing that our scanner can do right out of the box. So this is a technique that's really useful for looking at the vascular system in the brain. So these are just two images I pulled up. Where you can tell there's just, you know, versus normal MRI, you can see like extremely great resolution of just the vascular system. And so it's interesting to use these techniques in disorders that are known to have vascular deficits like diabetes and other conditions. So other more broad things that our scanner has that are relatively unique. The scanner is capable of what's called multi-band imaging or multi-slice. So typically in MRI, data is acquired slice by slice. So you can imagine like if we just say, particularly we're looking at the whole brain and we're acquiring in the axial plane. So you're taking slices out of the head one by one, and that's actually the signal acquisition. So some time ago, some scientists came up with an idea with a way of acquiring multiple slices. So instead of acquiring just one slice, you use different frequency compositions and you can acquire multiple slices all at the same time. And then you can actually tune this where you can acquire sometimes up to like 12 slices at the same time. So if you imagine that increases the acquisition rate of data exponentially. And ours, that's one of the great things about the Prisma is it's already set up where you can do that really to the edge of science. So this is, when applied to fMRI, it's often called fast echoplanar imaging or fMRI. And doing the task, you can acquire much, much higher data. So just as an example, historically, we would acquire data at like a TR of 3 seconds. So that means you really got one measurement every 3 seconds of the whole brain volume. Now you can pull that down to about 300 milliseconds and even a little faster. So then you're getting a whole brain volume of measurements every 300 milliseconds. So if you imagine the person's behaving, 3 seconds, we can do a lot of cognitive, actions in 3 seconds. 300 milliseconds is much more in line with, the way the brain's actually functioning. Now that being said, it's still measuring a blood flow signal, and blood flow is not super fast. So in a sense, you're oversampling the signal, but that helps in the fact that you have more data, better signal to noise, and so you can do a lot more with it. It also benefits diffusion imaging. So that really pretty diffusion map that I couldn't remember the directions on, that one was acquired, I think, with like 52 directions or something like that. So if you imagine the directions, you're acquiring images that tell you that so the more directions you acquire, the greater you can distinguish tracks that are going in slightly different directions. So if you go back 5 years, the state of the art was, in about 30 directions. So using multiband imaging, you can actually acquire in a reasonable amount of time up to like 180 directions. So you can get really fabulous images in 10 to 15 minutes. The multiband also is benefit really for almost everything. And if you're studying children, because children typically don't like being in MRIs, and so they do move a lot. So you can actually acquire data quite fast, and that's before they've done a lot of movement. It's also useful in applications like spectroscopy, just because, again, you can acquire more and more data in a smaller unit time. Our system also has research licenses where basically we can get other sequences that are not approved from other institutions. So like if you have a collaborator that's doing something very special that's not commercially available, we can sign a what's called a C2P agreement with that institution and then actually get that sequence on our scanner and then use it the same as them. Typically, it's an agreement that just says, like, the person who wrote it wants you to cite their paper or something to that degree. So it's not usually a big deal. Along those lines, we have a lot of major canned research protocols that are already on the scanner. So a huge study called the Human Connectome. We have those sequences on the scanner where you can collect the same data that was collected in the Human Connectome, or at least parts of the Human Connectome. Now the Human Connectome is a gigantic study with a lot of different parts. We also have the ABCD protocol, which is the largest neuroimaging study ever done in children. Ultimately, it will recruit 10,000 kids. The DevCog protocol, which was a large developmental study that is winding up now, that was a collaboration between 3 sites and started about 4 years ago. The megapress, which is a way of looking at local GABA concentration. So this one is currently was developed by researchers at the University of Minnesota, the flavor that we have, and it allows you to look at GABA concentration in relatively small voxels. And then, of course, we can add really anything we want. So if we want other things, basically it's just up to what the research community using the magnet wants. So what can you do with some of these things? Well, so this is a recent Nature paper that was based on the Human Connectome protocol. And what they did was they did an advanced analysis of several different sequences or scans, and were able to mathematically derive these functional regions that are kind of analogous to, if you're familiar with Brodmann areas, where they were based on histology. I forget, 120 years ago or 100 years ago. Very long time ago, and based on one brain. This is based on a gigantic sample. And the idea is that these brain regions tend to behave differently when the person's even just resting. So they're most likely different functional regions. When you look across, there's like 200 different ones. And so in these maps, like you can tell, like in the visual areas, this is the visual area unfolded. There's lots of different regions which map onto what we know about visual function in non-human primates, and to some degree even human fMRI at high fields. But so basically just creating extremely high resolution maps. These are all from controls, but of course it can be done in any population. So just the brain. So obviously I've talked about everything neuro-oriented. Everything I've talked about was neuro-oriented. That's more just because that's my training and specialty, but it's a full body scanner. So you can acquire anything using it. And we actually hope that people from different disciplines who are interested in the heart or different lung, different organs. I know someone we're already collaborating with is interested in pancreatic cancer. So the idea is like, you know, what can we do to take an extremely high resolution picture of the pancreas? This was actually something we did during training with the Siemens people here, and this is an image of the heart. Let's see if it plays. It's super, to me, very cool. I'm not a cardiologist, so maybe it's not, but it's actually the heart beating, and then down here is the EKG, and all this is MR acquired, so you almost have like a real-time representation of the heart while it's beating. But to me, it's really cool. Okay, so in contrast to MRI, the MEG is, it just measures magnetic fields. So you don't have, I would say, the latitude that the MRI has with the MEG. So basically, the signal in MEG is always going to be a magnetic field generated by active neurons. So just as you have electric current and power lines outside and magnetic field going around them, you also have electric current in activated neurons, and the magnetic field goes around them in a very predictable fashion. So basically, what you're doing here is you want to determine what the actual cells are responding to. So the way you do different things with MEG is that you administer different types of tasks, and the neurons that are responsible for allowing the person to execute that task become active, and so that's what's contributing to your signal. And so it's more of a problem of how do you identify the populations that are active. So a task that's pretty common in the MEG lab here. So there's basically lots of different sensory perceptual tasks. So looking at the visual system, auditory system, somatosensory system, motor control tasks. We have lots of different CAN tasks in the lab that were developed over the last 10 years. Working memory has been a very popular area for looking at the function of working memory networks using MEG. And then executive function and other domains. Really, there's tons and tons of possibilities. It's just it's determined by what the person actually wants to study. Of course, you can also look at resting state MEG and what we typically refer to as spontaneous. So when a person's just sitting still doing nothing, your cortex, the cells in there are just spontaneously firing, the spontaneous like population level discharges. And so historically, people didn't really know what those were about. Do they tell us anything about brain function? Are they just noise? In fact, my lab treated them as pure noise for the first 8 years of its existence. And then we actually started like looking at like patterns in that spontaneous activity and found really cool things that change as we age. And so now it's like a focus area for us. But MEG is unique in that it can actually see those spontaneous oscillations. And so that's one of the really unique things about the system. You can also, as with fMRI, derive resting state networks and then look at specific neurophysiological events. So MEG is used clinically to look at epileptic spikes and things like that. But there's really tons of different physiological events you can look at. So once you've actually made these populations, you know, and trained them in a task in some form or another, then you have to image them to get the pretty pictures. So our MEG lab here is equipped with lots of different functional mapping approaches. Beamforming is one of our specialties, but we also do lots of distributed solution models, minimum norm, Loretta, and different ways, basically of turning MEG signals into functional dynamic images. So just a quick, so beamforming, as I mentioned, is kind of one of our specialties. So here you're just taking the human brain, dividing it into hundreds of thousands of voxels, and then creating an image by computing the actual current density in each one of these voxels. Then that gives you a map that you can look at. So this is basically the outcome. So it looks at the end after doing all this very similar to a fMRI map where you have a statistical representation of what brain regions were active as the person was completing the given task. Minimum norm estimation is a similar technique. So this is basically the same thing, except the brain's been inflated and they've imaged activity during an auditory task. And you can see kind of what you would expect, activation in near the auditory cortex. Minimum norm's a little different in that it has specific assumptions about what brain activity what parameters it should follow. And so basically, if you're using that technique, you just have to bear the assumptions in mind. So one of the unique advantages of MEG, regardless of what imaging technique you use, is that you can look at the time series. So this is actually a time domain representation where you can see like very fast oscillations and very very strong response near time zero here, which was a motor response, person just tapping their finger. But so one of the with fMRI, as I mentioned, like state of the art is sampling the whole brain about every 3 seconds or so. That's every 300 milliseconds or so. By MEG standards, that's still quite slow, and neural events are happening millisecond by millisecond. So that's really MEG's specialty, is to capture that and provide a very highly precise representation of time. So you can look at the time domain signal like this, or you can also do what's called a time frequency decomposition and look at how different parts of the spectrum are being engaged as a person completes the task. So a way of kind of thinking of this is that sure, you have a time domain signal that you're looking at, but embedded, superimposed in that are lots of different activities occurring at different frequencies. And so you can pull that apart and image networks in the brain that are actually operating at within different spectral windows. So it's really gives you like insight on neuronal coding and just advanced neurophysiology. So just kind of a pathway of how these analyses typically look. So let's say you have the person's in this task is they're doing 2 different kinds of movement, complex and simple. And so you have them, completing this task, each of the different types of trials, complex and simple. You then image complex and image simple using one of the techniques I mentioned. Then you can do a statistical comparison between the 2 conditions, and you get brain regions that were more engaged in whichever condition. Here we had the prefrontal cortex and the parietal cortex that were more engaged during complex movements. You can then extract a time series from these clusters. That's what's plotted down here, with the different colors being complex in red and simple in blue, and see like when the 2 conditions actually differed as a function of time. You can then take that one step further and say, so what were these 2 brain regions? When were they talking to each other? So like, did that actually contribute to it? So what would be the correlation of neural activity in this region and this region? And that's plotted here, again, with movement onset being 0. And you can see that the interaction between the dorsolateral prefrontal cortex and right parietal cortex sharply increased right as the person was conducting the movement. So briefly, just some differences among the modalities. So I kind of alluded to this earlier. MRI is really multiple modalities. So I know, like, often it's said like, well, there's MRI and there's MEG, but MRI really is a bunch of different modalities all rolled into one machine. And that's an important thing to realize. So like when you look at an MRI scanner, nowadays, basically every MRI scanner can do functional MRI and diffusion MRI and structural MRI. It's not like there's special devices. It's almost all of them in the world now can do. All of them that were installed in the last 10 years can do it. Functional MRI. So first, there's high resolution brain structure. So here you're basically just looking at the structure of the brain with and high precision. Functional MRI, which is more what we talked about, where you're looking at blood flow changes as a function of the task and different regions becoming engaged. You can also do that in the resting state. Diffusion MRI would be a different modality. Again, you're looking at white matter tracks. Then spectroscopy, you're looking at metabolites. And then perfusion, just looking at blood flow, quantitative blood flow. And then you can also look at other things with MRI. MEG, in contrast, is a functional measurement only. So basically, you're just looking directly at the neurophysiology. Benefits of that is that it's not tied to a vascular signal at all. So like if you have patients with, say, diabetes who have vascular problems, sometimes their fMRI is going to be biased one way or another because of those vascular problems, whereas their MEG should not be affected to the same extent. Benefits of MEG, as I mentioned, has very high temporal precision, which is needed to see oscillations and spectral coding. It also has great spatial precision in the cortex. So in the neocortex, it's roughly equivalent to fMRI. And as you go deeper, fMRI gets superior in how spatially precise it is. Practical differences, MEG is far less claustrophobic and it's totally silent, so the system makes no sound. In our experience, it's tolerated by about 98% of people. We have very few, we've had huge studies here, and very seldomly are people not able to complete the procedure, because you're sitting in a pretty large room. There's nothing over your face, so it's actually relatively pleasant. On the other hand, the MRI is very noisy, and it can be claustrophobic, kind of, just depending on the particular person. It's tolerated by probably about 90% of people. So we do have quite a few people who either decline the MRI, or once they're there, decide they really wish they hadn't volunteered. And then MEG is also far more child-friendly. So there's groups that are really good with kids that go down to 4 years and younger, doing MEG scans while the kids are awake and behaving. So you can really do a lot with kids using MEG. So I'll just briefly touch on some of the things that are being done. So I mentioned structural imaging. We have ongoing studies, or there are ongoing studies in both aging and child development. And so one of the cool things, so as I mentioned, you can get very great precision with our magnet. And so you can look at just the gray matter, so the very thin layer on top of the brain, and you can look at how the thickness of that changes as a function of age or as a function of development. So it's typically from about 5 years on getting thinner in most regions as synaptic pruning early in life, and then later in life degeneration. So these are just maps of that, looking at changes as a function of DNA methylation age, which is you can also look at chronological age, but DNA methylation is just more of an epigenetic, thought to be a more precise measure of chronological age. But just looking at how the brain changes as a function of age and as a function of development. There's the resting state networks that I talked about earlier. So there's lots of studies looking at those that are currently ongoing here, and how the different networks are interacting. There's about to be a spinal cord study start that's looking at the disruption of white matter tracts in the spinal cord in kids with cerebral palsy. There's been lots of research already ongoing in cerebral palsy, but using our new system, we can do some really cool stuff with the spinal cord. So we're now starting that. High directional brain diffusion. So there's been ongoing studies in brain diffusion in typical development, healthy aging, HIV infection, chronic cannabis. Again, just looking at, are white matter pathways disrupted in these diseases, and how do they change as we age or develop, depending on the age group. Oops, I skipped one. Nope. And then other MR imaging studies. So there's lots of studies that are currently using the perfusion application of the scanner in both HIV, aging, and stroke. Multiple studies getting started now that are looking at GABA spectroscopy, including children with hearing loss and aging. And then new studies starting that are using susceptibility weighted imaging. We're using that in our new aging with HIV study, and also planning to use it in a diabetes study that's starting very soon. In regards to MEG imaging, so basically it's more task-based. So we have lots of stuff looking at visual attention. This is a study that was published last year by a student at UNMC, looking at whether you could use the MEG to distinguish HIV-infected patients who were cognitively impaired from those who were not impaired. And the impaired group here is his hand. So looking at just the amplitude of these brain responses during a visual attention task. Also looking at somatosensory function and how it changes as a function of aging. Just looking at whether somatosensory networks and somatosensory responses, whether they get weaker as we age, and whether the response to repeated stimulation varies. You can also use the scanner to look at the effects of treatment. So this is actually a study where we're applying brain stimulation to healthy controls and looking at whether that increased their cognitive performance or decreased it. And at the same time, we were looking at how those increases and decreases were connected to the neurophysiology in different brain regions. So having participants undergo MEG either after a sham stimulation session or a active stimulation session. And then studies using MEG in diabetes patients. So there's been some studies here looking at whether you can look at different neurophysiological events in the brain and connect those to clinical markers such as how long they've had diabetes, or their hemoglobin A1C measurement, and looking at like how well they manage their diabetes. Does that correlate with brain function in different areas? And then there's some large studies now looking at both drug and alcohol abuse and how that is reflected in the brain. This one in particular was looking at heavy chronic alcohol use versus low alcohol use being the control group, and looking at whether just occipital responses to basic visual stimulation differed in those groups. And if it differed, then what like time series. So shown here is just the actual time series of these occipital responses with the visual stimulation happening at time 0. So you can see like major differences later in the process. And then of course there's lots of normative work. So one of the cool things about MEG is that we're still only one of about 35 centers in the nation that has one. And ours is state of the art. So we're able to still look at just how healthy brain function works. So bringing in healthy controls, often undergraduates, and looking at how they're able to complete this task, how that's implemented in the brain. But that's still probably at least 40% of the studies that are done here. And they do quite well. And then I didn't have a lot of time to go over it, but there's a lot of other people who are using the scanners at UNMC and beyond, looking at different neurological disorders, different psychiatric disorders, and then how other problems in the body affect the brain. So finally, I'll just touch on some of the access. So scheduling the new magnet. So it's scheduled through RSS, which basically everyone has access to. Everyone at UNMC has access to RSS. Current rate is $600 an hour with 30-minute additional increments afterward. But an asterisk there, because that's kind of an ongoing discussion. We just now got started. So whether we want to keep the minimum being an hour and the 30-minute additional increments is currently being debated. But that'd be our policy for at least the next year. The usage fee includes a basic neuroradiology overread. Users can see the whole calendar and reserve times without assistance. So basically, you already have RSS, and then if you express interest in scheduling the scanner, then we give you special privileges, and then you can just reserve the scanner at your leisure. And then temporary data storage is also included. So once your data is recorded with the scanner, we push it to a UNMC-backed server where it's safe until you're able to move it to your personal location. The core is managed by Lisa Hausman. So this is Lisa here. She also operates the instrument, and then she's has 10 years of experience with MRI and is very sharp. So she's definitely someone who's available to help with protocol development and design consulting. I'm also willing to help. And then if you're getting something that's more advanced than us, I can help connect you with other people in the field who can help you with your particular application. The MEG system, it's scheduled through Outlook calendar. Current rate is $621 an hour. There's no neuroradiology overread needed because there's nothing really, we don't know what abnormal is with the MEG yet, so no reason to look. Users can see the whole calendar and reserve times without assistance. Again, we just give you access to it and go in and reserve it as needed. And we also provide temporary data storage once the data is collected. The MEG lab is managed by Nicole Knott, who also operates the system. This is Nicole. And for protocol development assistance and design consulting, I'm probably the best contact. MEG can be a little more temperamental, and it's not as it's not as fine-tuned as the MRI. MEGs, since there's only 35 in the country, they're quite temperamental still, so it takes a little more TLC. But finish there. So any questions? So that's the scanner is basically you can schedule it anytime you want during the day. If you're going into the afternoons or the weekend, we schedule them as well. Just have to let us know that's what you want so we can make adjustments.
I know you described for research, but I wondered, do you have any examples yet of, for example, with MEG, how it might be used in clinical practice today somewhere?
Sorry.
I keep thinking about when a person had a stroke now what maybe some of these instruments can do to help determine the ability to rehabilitate stroke victims also. But anyway, I'm just wondering, what do you see as moving forward, particularly in adults?
So at least for MEG, so currently MEG is used clinically for functional mapping of epileptic tissue. So patients who have intractable epilepsy who are going to undergo surgery, they'll come into the MEG center clinically, and we can use it to identify the tissue generating their seizures. And then we can also map out the eloquent cortices, so like where in the brain language is represented, motor function is represented, and so on. And then all that's passed in neurosurgery. As far as beyond that, it's also used in tumor patients who are going to undergo surgery. Probably the next possible use would actually be diagnostic. So there's a lot of groups trying to find biomarkers using MEG where they could then say, oh, this person definitively has autism or schizophrenia or something like that. Some of our HIV work is actually geared toward that, where we could identify those patients who are cognitively impaired versus those who are not based on this test alone. But I think there's a lot more in regards to just developing applications in the area of MRI. So there's lots of like major interest right now in eliminating contrast agents. So using some of the special like sequences that can be designed to basically get the same sort of precision in diagnostics without contrast as with. And particularly because gadolinium and other contrast agents are starting to have a more negative connotation with them.
Thank you.
You can repeat this question too, so people online.
Oh, yeah.
The MR spec is really interesting to me. So besides GABA, are there other metabolites people have been interested in? And what other disease states this sort of proliferated in? And has it gone beyond the brain? I'm curious.
So the question was about MR spectroscopy and what other metabolites can it see, and has it went beyond the brain? So a couple of things. So MR spectroscopy is like has a really dirty history. So it's actually was, to my knowledge, done back in the early 1990s. And the problem back then was that it was more of approach from a purely physics perspective, where can you look at these metabolites? And so the voxel was gigantic. I remember when I was a graduate student at Minnesota, we were doing spectroscopy, and we would take I think it was 5 centimeters by 5 centimeters, and we would just put it right in the middle of the head. And there was no real care put into whether it included white matter or gray matter or anything. But back then you needed a voxel that size to actually get a decent signal to noise ratio. And so it got the rap of being highly unreliable because there's so many things inside that voxel. And the more recently, so kind of fast forwarding to the last 5 years, been a lot of people who became very interested in it. And so now you see where it's improved a lot. The voxels are quite smaller. In regards to the metabolites, I have to go back and look. So there's actually big tables you can pull up about what some of these metabolites have been connected to. So each one of these, so basically the way they're interpreted, like creatine, they do different ratios, and then they've done studies to, because this is the spectrum here, they've done studies to connect different ratios of these metabolites to different processes in the brain. So that's like, there's several I know that have been connected to inflammation. And I think the way they do it is they do it in animals, and then they can go in and look at the actual histology and pull out what's going on. Yeah, beyond GABA, I know actually not that much about spectroscopy, just because that's kind of what we've always looked for. There's also ways you can look at glutamate, but that one's more controversial now, and a lot of people are arguing about how specific that signal is to glutamate, and glutamine. Whether it's moved beyond the brain. So I don't know, I'm so neuro-oriented. I haven't seen it anywhere else, but there's no reason why you couldn't do it in other organs. Yes, so data from our scanner, it depends on the actual study. So the DEPCOG study, which I briefly mentioned, is a 4-year longitudinal study of kids. All of that data will be publicly available in, I think, like 3 months with the conclusion of the study. And it will be available through Coins, which is a system you can get access to. And before then, we also allow it to go out to interested PIs just by emailing me. And then other studies, most of our studies that occur on the scanner are NIH funded. And so nowadays, most of the NIH funded studies, part of the actual award is that the data is shared at the end of the study. So almost all of it either is in the public domain or will be. And largely it's through the NIMH data archive.
Yeah, thanks for the presentation. I have 2 questions. One, there have been some more and more literature of the kind of using like integrating interactive model of using MAG and FMR at the same time for the same cohort. So I wonder if you have any thoughts on that.
Yeah, so first question was that there's been a lot of interest in combining MEG and MRI in the same study, and whether we have interest in doing that. So I'll go ahead and answer that one. So yeah, we definitely have a lot of interest in doing it. The fMRI and MEG are very complementary. Deep in the brain, fMRI has superior spatial resolution. Superficially in the brain and the cortex, they have more equal, but MEG has much more precise temporal resolution. So combining them makes a lot of sense. And we have a couple now of ongoing studies where we're combining them.
The second question is, as you know, I'm a neuro researcher at the same time. As you may know, especially for the last, like, say, 5, 6 years, there's a huge inflation of the end, the number for each modality. Like MRI, back in the days, you had like 15, now it's, let's say, 100.
Right.
And as a clinician, how to say, it's kind of getting further away from clinical utility because it's getting away from the individual. Meaning, if you need like 20 patients of anemia to see something different in hemoglobin from normal people, you can't do it in a clinical sense, right? So, as a clinician, that kind of inflation of the end, it's sort of like it's getting away from the clinical utility in my sense. I wonder what.
So the question was, it seems that neuroimaging, the sample sizes are growing exponentially, and that's making the technique less and less applicable to patient studies, where you often need more individualized conclusions. So yes, I think partly what's driving that is the emphasis on rigor and reproducibility at the NIH. So I'm on a study section that we only do neuroimaging, and so I would definitely say that the NIH is pushing for larger sample sizes, and they want large studies that provide definitive answers. That being said, I mean, I would agree with the idea that an effect you found in 12 people when you replicated in 90 people, I'm not sure the value added there. I think partly the idea, at least I see this on the review side and program interest, part of the idea now is that you have these gigantic samples, and then you're able to find fingerprints of diseases using these samples because you have much greater power. And then that at the end, you would have these fingerprints, and then you could take it back to the individual patient. And then you would have more of a diagnostic instrument based on, because you would have more of a space where you could actually, oh, this person falls in this, this little band, so therefore they have this disorder or this severity. But yeah, I mean, I think it's being driven by NIH currently. And then of course, journal editors are responding to that because they're getting more and more studies that have these huge ends, and so they emphasize those. Good question.
I think that it was a great presentation. In the beginning, you showed us the setup of MEG versus the MRI, quite different the MEG that is kind of sitting.
Right.
Give more availability for some executive function that task. So my question is that, as a wonder that means that as long as subject will fit into the helmet, they can do whatever they want. The researcher interested about movement analysis, you can develop whatever movement type you want, as long as it's feeding that seat. Or there's still a limitation, like the movement artifact at one extent of movement that we are not allowed to do, for example, walk, things like that.
Right.
Can you give some suggestions?
So the question was that from the images of the MEG system, it looked like the person's seated. And so is there any limitations in regards to what type of movements they could perform while seated, and what kind of degrades the signal? So the answer is, there's an investigator on campus who's been using the system for 8 years, Dr. Kurz. So he's actually developed lots of different tools over the years to record all sorts of different, like, leg and ankle and knee. And at one period of time, he actually developed a bicycle with the help of UNL engineering, and we tried to implement that. The bicycle didn't work. There was too much movement, but all of his other stuff has worked. I would say the only real concern is that, like, if the person's moving their leg, especially if they're lower functioning, often when they move their leg, they move their head. And this is even people who are high functioning. I've noticed, like, because I've been in the scanner lots of times, if I'm moving, like, my toe, I have a tendency to move my head at the same time. And of course, that's, what we're against, what we don't want. And so that's really, like, the only concern is, like, when if they're making a movement that is also going to create a head movement, and that's a concern. But there's ways of, Dr. Kurz has also designed, like, a bladder system where he actually can put a bladder up there and make the head more stable. So then you can actually move your head more, and that gives you some more movement. But yeah, if you're interested in that, connecting with him, he's done a lot of different things that no one else in the country's done in regards to, like, movements in the MEG. Yeah, okay. So you can get DTI. So the new scanners in Buffett, so they have a 32-channel head coil, and you can get pretty good diffusion imaging on those. The Philips 3T that's in Hickson Lead, it has an 8-channel head coil, and you can do diffusion on those, but I think the peak is 32 directions, and that's really not adequate these days. So yeah, you can definitely, the Philips and Buffett, they do pretty good diffusion.
I think we're going to end it there. Tony, thanks so much.
Sure, pleasure.